Hello everyone, welcome to today's webinar. I'm Catherine Dold with CNEN and I'll be moderating today's event. This webinar is titled Discovery and Characterization of Oligonucleotide Therapeutics and is being sponsored by WuXi AppTec. CNEN works with sponsors to identify topics of interest and value to CNEN's audience and consistent with their mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look for the Help widget at the bottom of the screen or type your query into the Q&A box. If you get disconnected during the webcast, please log back in according to the instructions you received earlier. You're encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. Our speakers will answer them at the end of the presentation, and as your moderator, I'll be posing as many as time permits. Any questions that we don't get to today will be answered by email after the event. Please note that CNEN does not endorse any company products or services that may be mentioned in the webinar, and that the webinar will be archived at CNEN online after the live webcast. The presentation today is being sponsored by WuXi AppTec. WuXi AppTec provides a broad portfolio of R&D and manufacturing services that enable the global pharmaceutical and healthcare industries to advance discoveries and deliver groundbreaking treatments to patients. Through its unique business models, WuXi AppTec's integrated end-to-end services include chemistry drug CRDMO, biology discovery, preclinical testing and clinical research services, and cell and gene therapies CTDMO, all helping customers improve the productivity of advancing healthcare products through cost-effective and efficient solutions. Our speakers today are Dave Madge, Chung Zhou, Jun Zhou, and Nan Zhao, all from WuXi AppTec. I'll now hand the program over to our first speaker, Dave Madge.
Hi everyone, and welcome to this webinar on oligonucleotide drug discovery and development. My name is Dave Madge, and I work in the Discovery Services team at WuXi AppTec. We're a contract research, development, and manufacturing organization supporting many modalities, and oligonucleotide therapeutics has been a major area of investment and expansion for us in recent years. In fact, the range of modalities available to us in drug discovery continues to evolve and provide us with ever more opportunities to explore the relationship between disease, pathway, and the most appropriate modality for therapeutic intervention. In this regard, nucleic acid-based molecules have come to the fore recently, especially in recent years as we have made significant inroads into understanding how to make such molecules stable to endonucleases and exonucleases, and how to modify for improved efficacy and selectivity. In this webinar, I'll say a few words by way of introduction to the area, and then I'll hand over to my colleagues to provide a detailed description of their respective platforms. In particular, Chung Zhou from our biology team will describe how we have built an integrated biology platform for the characterization of oligonucleotide therapeutics. Jun Zhao will describe our capabilities for the synthesis, modification, and conjugation of these molecules. And Nan Zhao from our DMPK team will talk about strategies for quantifying oligonucleotides and their stability and distribution. It's clear that nucleic acid therapeutics are already having substantial clinical impact, and the number of approved drugs and molecules in the later stages of discovery is rising fast, as depicted on this slide. With the advent of long-term clinical data for some of these molecules, including multi-year studies with molecules such as inclisiran, an siRNA directed at the mRNA of a target called PCSK9, we now know that effective suppression of protein synthesis by this sort of molecule can be achieved with dosing frequencies as long as every 6 months, which is extremely beneficial for long-term control of metabolic disorders, for example. This slide shows some of the diverse ways in which nucleic acid therapeutics have the potential to modify mRNA translation in particular. We don't only have opportunities to modify this process at different stages or by interfering with different elements of the process. We also have different types of therapeutics we can use, such as aptamers, short-activating or short-interfering RNA, antisense RNA, and others. Each of these have their own discrete applications and their own advantages and disadvantages, which gives us a great opportunity to match the choice of therapeutic mechanism to our understanding of the pharmacology and molecular biology of the disease and match this to the right therapeutic strategy. In fact, these different types of strategy also differ in their structure and topology, as shown on this slide. So we have molecules here composed of single strands with an extra mismatch sequence, and other variations, each of which have subtle differences in the way they work and the functional consequences of their activity. In this webinar, we'll focus largely on short-interfering double-stranded RNA therapeutics, but many of the chemistry, biology, and DMPK techniques we will discuss are transferable across these different formats. This slide provides a high-level overview of the workflow we've built for selecting an initial set of siRNA molecules to probe for efficacy at a new target. Briefly, the bioinformatics workflow looks at the sequence of the target gene and creates a set of candidate possible siRNA sequences for that gene. These sequences will be further ranked for a number of key features, such as strength of on-target and off-target activity, overlap with SNPs, match the different isoforms, and across species, as well as novelty. In this way, we can identify a highly ranked set of perhaps 100 or so sequences for synthesis and testing. Of course, even with the sequence in hand, we will need to make some strategic decisions regarding the topology of the double-stranded sequence we make. And we show some of the options here, as well as choosing the crucial modifications we'll make to the building blocks of the sequence and the bonds used to connect those building blocks to optimize for stability in particular. Then finally, we'll consider how we will deliver our sequence, whether we use conjugation to the ASGPR receptor ligand like GalNAc, for example, or whether we use a lipid nanoparticle or some other delivery mechanism. On this slide, we show some of the more frequently used chemical modifications to the building blocks of a nucleic acid therapeutic, and how these have developed and become more optimized through several iterations in recent years. First-generation compounds had fairly simple changes, such as the use of phosphoramidite linkages, while second-generation modifications included the adoption of widespread substitution of the 2' position with fluorine or methoxy, for example. More recently characterized modifications include additional ring closures on the ribose and morpholene replacement of the ribose. Jim will talk more about the synthesis and application of these modifications. So to wrap up this introductory section, what you will hear about over the next 30 minutes or so are some of the key elements of the integrated oligonucleotide discovery workflow that you can see depicted on this slide. We've built and validated tools to support target selection and validation, sequence selection, synthesis, in vitro testing and in vivo testing, as well as the optimization, toxicology, and manufacturing of this exciting new class of therapeutics. I hope you find this webinar interesting. Please do feel free to contact me or any of the speakers for more information. Many thanks.
Hello, everyone. This is Chung Zhou from Discovery Biology, WuXi Uptake. I'm glad to have this opportunity to introduce our oligonucleotide biology platform. Oligonucleotide meaning included antisense oligo, siRNA, microRNA, aptamer. There are more than 10 oligo drugs marketed so far. Since antisense oligo and siRNA interact with the target RNAs with the complementary base pairing mechanism, it is convenient to use computer programs to design oligo sequence. Theoretically, all the RNAs can be targeted, including the traditional undruggable targets for the small molecule and antibody drugs. Therefore, the oligonucleotide is considered to be the third major therapeutic modality. The advances of antisense oligo and siRNA drugs mainly are attributed to the breakthrough of the chemical modification and the delivery methods. Chemical modification can increase the oligo stability, decrease immune toxicity, and off-target effects. The delivery tools can improve the organ targeting efficiency, especially GalNAc can specifically and efficiently target hepatocyte. Currently, there are a lot of researches to explore the actual hepatic delivery of oligos to treat the other disease. So similar to the discovery of the other type of drugs, for oligonucleotide drug discovery, we need to improve the potency and decrease the toxicity of the compound. Our service cover in vitro, in vivo activity evaluation, delivery efficiency, and early tox, including the immune tox and off-target effects, and the others. The first step for the oligo drug discovery usually is sequence design. This is a flow chart of our siRNA design pipeline. We also have a commercial database can filter out the label or label the patented sequence. For antisense oligo, we are also building up the capabilities, and hopefully we can provide the service later this year. Next step is for the oligo drug discovery usually is to evaluate the compound activity in cell-based assays, in the transfection assays. So we have more than 400 cell lines in WuXi Biology Bank. Also, we can access to some patient-derived fibroblasts and some primary cells. So we can choose the optimal cells for the compound activity evaluation. For antisense oligo, there are two major mechanisms. One is to degrade the mRNA through the RNase dependent degradation mechanism. The other one is to regulate the splicing. So we may use some patient-derived cells for the splicing mechanism antisense oligo compound. For the mRNA degradation, ASO and the siRNA compound, we can detect the mRNA expression level by RT-PCR or the branch DNA. And for the splicing, we can use the capillary electrophoresis or Sanger sequencing and NGS sequencing to evaluate the intra-axonal inclusion and exclusion effect. And next, we may need to evaluate the protein levels by different methods. And then for some immature target, we need to do some cell-based function assays. We can provide the customer service for this part. And also, I want to mention that for some targets, the expression level is very low in the cell lines or the primary cells. We may need to use an alternative method, PCR check reporter. And in this reporter assay, the targeted mRNA is inserted after the stop codon of the reporter gene, and the reporter activity can represent the chimeric mRNA level. So for the targets with very low expression level in cell lines, the reporter is a very useful tool. And the assay is also lower cost compared with the RT-PCR or branch DNA method. Besides, the PCR check reporter is also useful for evaluation of the off-target effect. Currently, we have validated a lot of in vitro screening assays for more, up to now more than 50 targets. Here are some example data of the RT-PCR assay in a cell line by transfection of the compound. In the primary screening, usually we recommend to do 4 concentration or 3 concentration, and then select a compound with good activity to do those response curve EC50 determinations. And I mentioned PCR check reporter is an alternative method for the compound evaluation. Here, we also put some example data for the reporter system. You can see, in generally, the reporter system has the consistent data compared with the RT-PCR method. And the data variation and the reproducibility is acceptable. Since the oligos are relatively large and elective charged, so it is very critical to deliver the compound into cells. For the 5 marketed siRNAs, 1 was delivered by LNP, the other 4 was delivered by GalNAc conjugate technology. So it is very critical for oligo compound to find out efficient delivery method. For example, use the GalNAc LNP, PNP, lipid, or the peptide conjugation. We can provide a service to improve check the cellular delivery effect. Taking the GalNAc conjugation method as an example, we develop cell-based binding and uptake assays. GalNAc can bind to the SPGR receptor on the cell membrane and induce the receptor mediated endocytosis. We use the fluorescence labeled oligo compound and incubate the compound with the different type of cells. You can see that the HepT2 and the 293A cells, the GalNAc SPGR binding activity is very low. However, the fresh primary mouse hepatocyte and the cryopreserved primary human hepatocyte has much higher SPGR binding activity. So furthermore, we use the primary human hepatocyte to develop a competition method to evaluate the compound activity of the novel GalNAc compound. So for the other, next we may need to do some free uptake assays, which means we treat the compound with the cells without transfection and detect the target mRNA or protein levels. Then we can evaluate the combined efficiency of the oligo sequence and the delivery tools. So since in the cell lines, the GalNAc uptake activity is very low. So for this free uptake assay, we need to use the primary cells. Here we show some example in the primary human hepatocyte and the primary cyno monkey hepatocyte. And also we can isolate some fresh mouse hepatocyte from normal mouse or transgenic mouse or pharmacology mouse models to some specific targets. So for the other delivery method, we can also set up method by using some optimal cell lines or primary cells. We also can do in vivo delivery efficiency evaluation for oligo compound, including such as the lab imaging system with the fluorescence labeled oligo in mouse for the biodistribution studies. And besides, we can also detect the oligo in plasma or in tissues by different methods, including LC-MS. And my colleague Nan will talk about LC-MS in details later. And in our team, here I would like to talk about, we use the stem loop PCR to determine the risk associated siRNA. So we use the AGO2 antibody to pull down the risk complex, and the siRNA associated with the AGO2 complex, risk complex can be detected by the stem loop RT-PCR method. The left panel is the results of the cell line validation results. And the red panel is the results from animal studies. We detected the total siRNA concentration in liver and also the risk-associated concentration. You can see that the kinetics of the two type of compound, the total concentration and the risk-associated concentration is different. For the total siRNA, the concentration decreased since 4 hours continuously. For the risk-associated siRNA, relatively it maintained stable from day 7 to the day 28. And in general, the kinetics of the risk-associated siRNA is more relevant with the clinical data. In clinical, siRNA compound, the GalNAc siRNA compound can be treated every 3 months or half a year. And also we can see that the siRNA, risk-associated siRNA concentration is much lower than the total value. So it is consistent with the literature report that the endosome release efficiency is very low. So to improve the endosome release efficiency is objective of to improve the compound potency. So this method for the risk PK evaluation method should be a very useful tool for the related researches. And next, I would like to introduce about our animal models for the oligo compound evaluation. Since antisense and ASO compound, the mechanism is used the complementary base pairing mechanism. So we need to consider the homology between the animals and the humans, especially for the compound. The similarity between the mouse and the human is very big. We need to consider to use the humanized mouse model for the compound activity evaluation. First, for the liver target, we can use a hydrodynamic injection mouse model. And hydrodynamic injection is to inject the plasmid DNA with the human genes into the animals by the tailwind within 5 seconds. And then it can result in the plasmid DNA entering into the hepatocyte. However, the expression of the transgene usually is transient. So this model can be used for the liver targeting, short-term compound evaluation. And the second model is the AAV mouse model. AAV is a widely used gene delivery tool, and the transgene expression level is very stable, as long as many years in the clinical. And in our labs, also we observed it is stable more than 10 months. And by using different type of AAV, we can target the different organs. For example, AAV8 for liver, AAV PHP.B is for CNS. So we can use AAV to introduce the human gene in different organ of the mouse for different targeting oligos. And also, the model is useful for the long-term compound evaluation. The third model is transgenic model. Usually we prefer to use lock-in model. And by using the transgenic model, not only we can detect the compound activity on the target gene, target protein, and the biomarkers, also we can use the transgenic mice for the pharmacology model establishment. However, usually it takes a long time to get the animals. For the liver targeting, we can also use liver humanized model, and the liver human ratio can be higher than 30 or 90%. The advantage of this model is it is useful for the on-target and also off-target, because all the hepatocytes are human genes. But the cost may be much higher than the other models. In each project, we may need to choose the suitable model, may combine different models at different stages. So we can discuss with the client, share our experience and opinions for each specific project. In this slide, I show some example data for the HDI mouse. The left figure is the lab imaging of the BABC mice hydrodynamic injection with the luciferase plasmid DNA. You can see that the luciferase activity decreased very quickly. So usually for the HDI model, we pre-dose the animals and then inject the plasmid DNA. And this is a transient model. However, the data have acceptable and good reproducibility. You can see the data. The second study, the 3 doses showed very good dose-dependent effect. This slide, we put some example data of transgenic mice and the AAV mouse model. I just want to mention that for transgenic mice, we need to confirm the sequence before we use it, especially, for example, confirm whether the UTR sequence is included. And for the AAV model, I want to see that we can provide the AAV generation in-house to support the model establishment. Except for the mouse models, also we can provide the large animal services. So we put some example data here. For monkey, we can use different various administration methods, including normal IV, assay injection, and also some special injection, for example, IT, ICV, and so on. We can also do the liver biopsy to collect samples. And also we have very comprehensive post-lab analysis platform to support the PD study in both the monkey and the mouse studies. At last, I would like to briefly talk about our capability of the off-target and early tox studies. We can access the general cytotoxicity and also to evaluate the immune toxicity. Oligo compound can activate the toll-like receptors, RIG-I, MAD5 pathways. So we can use the reporter genes to evaluate the compound specific activity on these pathways. And also we can detect the downstream cytokines of these pathways, usually in human PBMCs, but also we can do some animal immune cells, for example, PBMC from monkeys. Moreover, for the sequence dependent off-target is a major cause for the toxicity. So we also have RNA-seq platforms can do some RNA-seq analysis to support the off-target evaluation. Usually we provide the standard analysis, data analysis, including differential gene expression profiles, and also we can do some sequence-based off-target evaluation. Besides the biology support for the oligo discovery, we can work with other WuXi BUs to provide integrated service. So my colleagues in the other BUs will introduce the other WuXi capabilities of the oligo discovery. So next, my colleague Jun Zhou from the chemistry team will talk about the chemistry part. Thank you for your attention.
Welcome to the webinar. My name is Jun Zhou, joining from WuXi AppTech. Today, I'm going to talk about oligonucleotide chemical modification and conjugation. Chemical synthesis of therapeutic oligonucleotides relies on solid phase synthesis using phosphoramidite chemistry. The synthesis typically starts from CpG solid support, onto which the phosphoramidite building blocks are sequentially coupled until a full-length oligo is formed. This process has been fully automated using a variety of oligosynthesizers. At WuXi Chemistry, our facility is equipped with two major platforms, including Mermaid system for small-scale and library synthesis, and Atkala system for gram-scale and large-scale synthesis. To enable further conjugation or labeling, a reaction handle can be attached to the 5' end, for example, a amino modifier that allows for further amide coupling on 5' terminal. Upon completion of a sequence assembly, the oligo is released from the solid support using ammonium hydroxide. The cooled products are purified by reverse phase HPLC or an ion exchange chromatography to provide the desired oligos with high purity. In some cases, post-synthetic oligonucleotide conjugation is performed in solution phase to link with a different molecule. For example, amide coupling to attach a peptide as an active ester to the 5' amino modifier. The final oligo product is analyzed by UPLC and TOF mass for purity determination and mass identification. Are most widely used as antisense oligonucleotides or siRNA. Since unmodified RNA can easily undergo nuclease degradation, a wide range of modified nucleic acids have been incorporated into therapeutic oligonucleotides to improve the stability, to fine-tune the drug-like properties, and to enhance the delivery of oligonucleotides. For siRNA, in addition to 2' methoxy, 2' fluoro, the UNA, GNA, and 5-VP nucleotides are incorporated to the antisense strands. And for ASO, a broad range of breached nucleic acids are introduced to the flanking region of the gapmers. Conjugation to targeting ligands at 3' or 5' terminals provides the opportunity to deliver oligonucleotides to specific tissues and to increase the half-life and retention, as well as intracellular uptake. The targeting ligands could be a peptide or carbohydrate, for example, GalNAc, lipophilic moieties, PEG, or a small molecule. At WuXi AppTec, we provide a comprehensive collection of modification and conjugation, and all this can be pre-made at building blocks on shelf, ready to go for oligonucleotide and conjugation chemistries. GalNAc is of highest interest to conjugate with ASO or siRNA to deliver the oligonucleotide to liver hepatocytes while specific binding to ASGPR with high affinity. From a synthetic perspective, tri-GalNAc ligand can be attached to either 3' or 5' terminal of ASO or siRNA sense strands through a variety of conjugation chemistry. For 3' GalNAc conjugation, the primary option is to load the tri-GalNAc onto CpG solid support, which serve as a start point of oligoassembly. For 5' terminal conjugation, the tri-GalNAc can be introduced as a phosphoramidite on solid support or attached through amide coupling or click chemistry in solution phase. At WuXi Chemistry, we have a successful track record in delivering more than 3,000 GalNAc conjugated siRNA on a variety of scales and more than 30 novel GalNAc ligand synthesis. Lipic conjugation of oligonucleotide has proven to enhance nuclear stability, prolong half-life and retention, and to increase intracellular uptake. The lipid conjugation moieties include long-chain lipid acids and other lipophilic molecules such as cholesterol and tocopherol. These lipid moieties can be linked to 5' ends through phosphoramidite coupling on solid phase or post-synthetic conjugation, including amide coupling or click chemistry in solution phase. For 3' lipid conjugation, the lipid moiety can be preloaded to CpG solid support, which is similar to GalNAc conjugation. Peptide conjugation can enable cell penetration and cell targeting delivery of oligonucleotides to specific tissues. For example, cell penetrating peptides, also called CPP, that binds with negatively charged cell surface proteoglycans across the cellular membranes, increases cellular delivery of oligonucleotides. Cyclic IgD peptide is a tumor targeting ligand and thus can be used to deliver oligonucleotides to tumor cells for cancer therapy. Peptide can be conjugated to either 3' or 5' ends through a variety of conjugation chemistries, including click chemistry, thiol-maleimide conjugate addition, and amide coupling. Prior to conjugation, the peptide moiety can be modified by appropriate functional groups that enables the conjugation. At WuXi Chemistry, we have established a highly convergent and versatile dual conjugation platform wherein an extensive collection of reaction handles can be introduced to either 3' or 5' terminal of an oligonucleotide. And these reaction handles can enable a series of conjugation chemistry to link with a broad range of molecular modalities. Moreover, the selections of appropriate chemistries that are orthogonal to each other would allow for sequential conjugation with two different ligands at both ends. Take one example. An azide linker is installed onto the 3' end that allows for copper-free click chemistry to attach a library of BCN modified lipids. And on the other end, the reaction handle can be in a terminal alkyne for copper catalyzed click chemistry to introduce a fluorescent tag. Alternatively, the 5' end linker can also be an amino modifier that can react with the peptide carboxylic acid active ester or maleimide linker that would couple with the thiol-containing peptide. The dual conjugation strategy is highly convergent and enables rapid installation of various ligands at both ends. At WuXi Chemistry, we provide a one-stop solutions to early-stage discovery of therapeutic oligonucleotides, and our services include synthesis of special phosphoamidite monomers, various oligonucleotide modalities, and the best solutions to oligonucleotide conjugation.
So, hello everyone, this is Nanzhao from DMPK department at WuXi Ltd. I'm very glad to have this opportunity to share our DMPK strategies for oligonucleotide therapeutics. Firstly, I want to give a brief introduction about DMPK. The purpose of DMPK is to study the characteristic interaction of a drug and the body for its absorption, distribution, metabolism, and excretion. In other words, DMPK is to figure out how the compounds of interest get into the body and how they move within the body and how they get out of the body. It can help assess the safety and efficiency of a new therapy. The DMPK of a new therapy should be studied and evaluated throughout the whole process of drug development. It can significantly improve the successful rate of the translation of a preclinical result to clinical manifestation. Now let me go back to the oligonucleotides. Oligonucleotides are a unique class of compounds that share characteristics of both large and small molecule. They're like a large molecule drug because they have relatively high molecular weight and possess excellent target specificity. However, they also share many characteristics of small molecules, including synthetic production, relatively simple chemical structure, and inclusion of chemical modification to improve its performance. This identity brings its unique ADME characteristic. So oligonucleotides is usually administered by IV infusion and subcutaneous injection or direct injection. After absorption, oligo are distributed within minutes to hours and rapidly uptake into tissues, mainly in the kidney and liver. And their exposure is much higher than that in plasma. The half-life of oligonucleotides in my organs like liver or kidney is from weeks to months, but in plasma it only takes few hours. Oligonucleotides is mainly characterized by extranuclease and endonuclease, resulting in shortmers and eliminated through urine and feces. And then next, I want to share some information about how we study the distribution and metabolites of oligonucleotide. Okay, here. The distribution refers to the movement of a drug between intravascular and extravascular compartments of the body. Because the protein binding and unbound forms of drugs can affect their distribution in the body. So it suggests to conduct in vitro protein binding study in early discovery. Ultrafiltration, ultracentrifuge, and electrophoric mobility shift assay are always used to determine the protein binding characteristic of oligonucleotides. For in vitro study, the biodistribution is requested not only for the oligonucleotides, but also for its delivery system, or the individual components of the delivery system, and the combined final product like the protein. The biodistribution data is always obtained by either analyze the main tissues separately or based on the radiolabeling or image for the whole body. And then it's about the metabolism. Different from small molecule, oligonucleotides is mainly catalyzed by extranuclease and endonuclease, resulting in shortmers. The shortmers may be pharmacologically active and interference with bioanalytical assays. Therefore, it's also important to investigate the in vitro and in vivo metabolites during the preclinical stage or discovery stage. Here is the workflow of metabolic stability assay. For the in vitro study, the sample was obtained by incubating oligonucleotides within liver isolate, hepatocyte, or liver homogenate from different species. While for the in vivo study, the sample was obtained from dosed animals. All the samples were extracted and analyzed by high-resolution mass spec, and then potential metabolite could be identified by using the software such as the Biopharm Finder. As a consequence, the in vitro and the in vivo metabolic pathway will be found. Here is summary of the key in vitro and in vivo ADME studies of oligonucleotide in the preclinical stage. It shows that the quantitative analysis of oligonucleotides from different assay types involves a variety of biological matrix and the target analyte. The target analyte is not only the oligo of interest, but also the oligonucleotide that may found as metabolites. In addition, the propagate of pharmacodynamic biomarkers, such as target mRNA, target protein, or downstream biomarker that reflects the modulation of the target protein, should be also measured. Therefore, it's important to develop and validate a proper bioanalytical assays to support oligonucleotide quantitation in biological matrix. However, the bioanalysis of oligonucleotide faced many challenges, requiring high laboratory platform construction and technical personnel expertise. As shown in the previous talk, the oligonucleotides has unique physical chemical properties like its size, diverse chemical modification, and polyanic nature. It requires advanced and unique analytical strategies. During the method development of bioanalysis, many factors need to be considered, such as the bridge between different platforms, multiple biological samples used for the PD and PK evaluation, and the interference from the impurity and metabolites that could interfere with the assay. Especially for the immunogenicity studies, the ligand binding assay always need to be optimized to avoid interference from the negative charge protein, the cross-reactivities of the anti-drug antibody to endogenous RNA and DNA. Many methods have been developed and validated for quantitation of oligonucleotides in vivo and in vitro ADME studies. The liquid chromatography mass spec and the ligand binding assays such as ELISA and qPCR are widely used for oligo quantitation. However, there are more and more techniques such as branch DNA, ddPCR, fluorescence illumination image, and bioillumination image applied in this area. They can be generally classified by 4 main categories and identify the oligo of interest by charge, mass, binding, or imaging. It's important to choose the correct assay, which can help to indicate by the individual molecular. The method selection should account for the chemical structure of the oligonucleotides, the requirement of the assay sensitivity, the sample type, and the throughput, and even the development stage of your study. Our DMPK department provides a robust bioanalysis solution to accelerate the development of oligonucleotides from the discovery to late clinical stage. So far, we have developed multiple reliable bioanalytical platforms with high sensitivity, selectivity, accuracy, precision, and throughput. There are LC mass spec, LC high-resolution mass spec, LC fluorescence detection, and ligand binding assay, and qPCR. Those platforms can support the oligonucleotide studies while viral stage. For example, the LC-MS method, which is that used for the small molecule, can help measure an oligo's concentration in target organs, such as the liver, kidney. This assay can achieve high specificity, accuracy, and precision. But as the program processes to later stage of preclinical and clinical studies, it needs more sensitive method to measure lower concentration in vivo samples. The ligand binding, such as the hybridization ELISA, MSD, or qPCR, are very helpful in this area. However, for this method, we may need to design the special probe and screening and synthesis this probe before we start the real method development. Okay, from the regulatory perspective, there is still some ambiguity for oligonucleotides. Although the US FDA considered oligonucleotides as a drug, not a biologist, should follow a regulatory framework of small molecular drugs for new drug application. However, the immunogenicity data, which is required for a biologist, was provided as part of the new drug application filing. It makes us to think about is which way we need to follow. Recent draft guidance from the FDA addresses the IND submission of individualized antisense oligonucleotide drug product, and also the clinical pharmacology consideration for the development of oligonucleotide therapeutics. With the development of oligonucleotide therapeutics, we believe in the future there will be more and more guidance released by the regulator for the oligonucleotides, and we will pay close attention to that. Okay. Our DMPK service department provides comprehensive in vitro and in vivo ADME solution from high throughput screening and in discovery through the drug development phases, including in vitro ADME, rodent PK, large animal PK, non-GLP bioanalysis, drug-drug interaction, metabolite profiling and identification, non-clinical mass balance, tissue distribution, and radiolabel human AME studies. Beyond DMPK, our skilled team and toxicologists provide safety assessment across a wide range of species and through various routes of administration. We also have a bioanalytical service team to provide and through various to support your drug discovery and a GLP compliance. We worked closely to offer a one-stop service to our client. Thank you for listening. Please visit our webpage or scan the QR code if you want to learn more about it.
Thank you for a great presentation. We have a few minutes left for questions. Just a reminder to our audience, you can submit your questions in the Q&A box at the bottom of your screen, and any questions that we don't get to today will be answered by email after the event. Let's get to our first question. How should we go about selecting humanized mouse models? What are the pros and cons of different approaches?
Thank you for the question. For selection of the humanized mouse model, we need to consider the stage of the program and also the target of the study. For the early stage discovery, we need to do the PD studies quickly. So we may need to choose the HDI, hydrodynamic injection mouse model, and as well as AAV mouse model for both the liver targeting and non-liver targeting disease. So by using these two types of model, we can establish a model very quickly. However, the model cannot be used for the disease model development, just can be used for the PD to evaluate the knockdown efficiency of the test articles. If we want to look at the long-term PD efficiency as well as some disease model pharmacology result, we need to use the transgenic mouse model. However, it will take a very long time to get the transgenic mouse model establishment. So we need to prepare the model in advance. And for humanized mouse model, which is just can be used for the liver targeting. However, for this type of model, we can use it for not only the on-target effect, can also be used for the off-target effect. So in summarize, we need to consider comprehensively for the stage of the study and the type of the target. Yeah, thank you.
Okay, thank you. Our next question, very impressive oligoconjugation capability for attachment of peptides to oligo on the 5' end. Would it be feasible to utilize phosphoromidite chemistry similar to GalNAc and lipid conjugation?
Thank you for the question. It's generally not recommended to go with phosphoramidite chemistry because peptide protecting groups are not quite as compatible with oligonucleotide cleavage and deprotection conditions, which might cause some issues at a very late stage. Peptide can be efficiently coupled to oligonucleotide through post-synthetic oligonucleotide conjugation in solution phase. For example, cleave chemistry and amide coupling. Thank you.
Thank you. And that is all the time we have today. Thank you again to our speakers for their fascinating presentations. And thank you to our participants for being a great audience, and On24 for technology and production services. And thank you, Wuxi Uptake, for the sponsorship that made this interactive webcast possible. Be sure to check CNN or CNN Online for information on upcoming webinars. For CNN Webinars, I'm Catherine Dold. Goodbye.